Welcome to this session. My name is Jouni Kuha. I'm currently the joint editor of JRSS Series A. And I say that because this whole session has to do with the journals. We have 3 papers that have come from the Society's 3 journals, A, B, C. The speaker's order is today BCA for some reason, but there you go. 3 excellent papers tied together by a theme. These are all on medical statistics, one way or another, which is, I think, highly appropriate. None of the journals are specifically about medical statistics, obviously. They're not biostatistics journals, but equally they're all about medical statistics, or can be, whether it be methods or applications of medical statistics. Those are quite obviously a big part of the kinds of statistics that the Society is engaged in. So it gives me great pleasure to host 3 talks on that kind of topic. As I said, we'll go Series B first. So our first speaker is Piao from University of Southern California, who with co-authors published a paper and gives a talk on the topic of semiparametric model for bivariate survival data subject to biased sampling. So without further ado.
Good afternoon, everyone. My name is Jean, and the topic of my talk today is semiparametric model and inference for bivariate survival data subject to biased sampling. This is joint work with Dr. Jin Ning and Dr. Yu Shen from UT MD Anderson Cancer Center. This is the outline of my talk today. Firstly, I will introduce some backgrounds of biased sampling. I will use a prevalent cohort of a breast cancer recurrence study from Sierra Medicare database as an example to show the challenges of analyzing such data. And we propose a modified EM algorithm to estimate the parameters of interest. Simulation studies are performed to evaluate the small sample performance of the proposed estimator, and then we apply the proposed method to the motivating study. What is the biased sampling? Biased sampling means that some members of the population are more likely to be sampled than the others, and there are some issues caused by the biased sampling. Including any statistics computed from the observed data may be erroneous, and we could over or underestimate the parameters of interest. For example, when the evidence combined from published study may subject to publication bias in meta-analysis. And how can we know if a sample is biased? A quick and easy method is to compare the characteristics in the observed data versus that in the target population. If they are quite different, it may indicate that the observed data do not present the target population. And also in practice, we may encounter biased sampling either by limitation of conditions or by design. The incident and prevalent sampling are to commonly used cohorts in epidemiology studies to study the natural progress of a disease. For incidence cohorts, we recruit a subject at the risk of the initial event, and then we observe the occurrence of the initial event, and then follow the patients until the occurrence of the event's interest, loss of follow-up, or end of study. The figure on the right panel showed the sampling mechanism. We can see that from incident cohort, we could obtain a random sample from the population. But when we deal with rare tumor disease, we may need to accrue many patients in order to have enough sample size, and also we may need a longer follow-up time in order to accumulate enough number of information. On the other hand, the prevalent cohort is a more efficient alternative. For prevalent cohort, we accrue patients already experience the initial event, and then we follow them prospectively for a subsequent event of interest. It can be noted that from the prevalent cohort sampling, we may require less patients and also shorter period of follow-up time. But the observed data may not represent the target population. For example, on the panel here, patients have to survive until the time point of enrollment in order to be included in the study. Then any patients with profile 4 will be excluded. And if we just analyze the observed cohort, we could have biased estimations. This is just a summary of the comparison from of the incidence versus prevalent cohort that I mentioned. And ignoring the sampling mechanism will lead to severely biased estimates from prevalent cohort. This is our motivating study, which is a study of breast cancer recurrence from Semiparametric database. It has been known that greater progress have been made due to early detection and also improvements in the targeted therapy. But despite the treatment advances, many patients may still relapse after their initial treatment. And for our motivating study, we identify a prevalent cohort from Semiparametric database consists of patients who have already experienced the initial event, which is cancer recurrences, is defined as having a subsequent mastectomy occurred more than 10 months from the initial conserving surgery. The identified cohorts, including 386 patients from year 2004 to year 2011. The primary goal of this study is to evaluate the association between risk factors and the residual survival time. Here, our primary endpoint is residual survival time calculated from the disease recurrence. And also, we want to predict the residual survival times. When analyzing such data, we have several challenges. Firstly, the observed cohort does not equal to the target population. This is due to the prevalent cohort sampling mechanism. As I showed earlier in the figure, if we just ignore the biased sampling, we would overly estimate the residual survival. And also, we need to think about, for this case, is this a standard left truncation data? And the answer is no, because the truncation probability would rely on the relationship between the overall survival and the truncation time, because patients have to survive until the truncation time in order to be enrolled into our study. And the primary endpoint of this study is residual survival time after cancer recurrence. It is also noted that there is positively relationship between our primary endpoint and the overall survival. So this relationship motivates us to jointly model the bivariate survival data. So here are some notations of the proposed method. We use, for the target population, we use U tilde and V tilde to represent the time from diagnosis to recurrence and the time from recurrence to death, respectively. And we use A tilde to represent the unbiased time from disease diagnosis to study entry. Due to the biased sampling mechanism, patients have to survive until the sample time to be included. So equivalently, it is the sum of tilde U and tilde V should be greater than tilde A here. And we use the characteristics without tilde to represent the observed biased counterparts. This figure shows the data structure of our study here. The bivariate survival time include the landmarks in the progression of a disease. So the U tilde here is from diagnosis to recurrence, and a V, which is our primary endpoint, from recurrence to event of interest. And under the bias sampling mechanism, any patients who died before enrollment will be excluded. So for this case, patients with profile 1, 2, and 3 will be included in the observed data, while patients with profile 4 will be excluded. And obviously, if we just analyze the observed data, we could overestimate the survival due to exclusion of a subgroup of patients. Here's our proposed models. For the marginal distributions, we use Cox model to model the time from disease diagnosis to recurrence, and also the time from the disease recurrence to death. And for the dependent structure, we use a Coppola model. Coppola model is widely used to model bivariate survival data because it has a nice feature that the marginal models and the dependent structure can be modeled and estimated independently. And for model identifiability, we assumed a parametric model for the truncation time, A tilde. It is not showing correctly for the formulas, but we can write down the likelihood. One intuitive thought is that we can just optimize the observed likelihood and to observe the MLE. But there are some computational challenges of maximizing the observed likelihood directly. For example, for this likelihood, it involves infinite dimensional parameters for the cumulative hazard functions, and also the association. There are associations between the bivariate survival times. And implementing the profile likelihood method is very challenging considering this form. So to overcome these challenges, we proposed a pseudo-likelihood equipped with a two-stage modified EM algorithm to estimate the parameters of interest. In the first step, we use conditional likelihood to estimate the parameters of interest for the truncation time distribution. And after that, we use the modified EM algorithms for the bivariate survival time. We treat the biased sampling problem as a missing data problem. And in the E step, we estimate the complete likelihood function. And for the M step, we motivated by the two-stage method by Xi and Luis, we estimated the marginal distributions and the dependent structures independently. So in the M1 step, we estimate the marginal distributions, and after that, we estimate the association parameters by fixing the margins at M step 1. And we can repeat E step and M step until convergence. The proposed method is very computational efficient because in the M1 step, we can use any existing softwares for analyzing survival data to get an update by just specifying a weight function here. And here are some theoretical properties of the proposed method. For theorem 1, we showed that the likelihood function of the observed data will increase with iterations. So it has nice ascent properties. And also we showed that the proposed estimators are consistent and will converge to a tight Gaussian process. More details regarding the proof can be found in our paper. Here is the reference. Then we performed the simulation studies to evaluate the finite sample performance of the proposed method. For marginal distributions, we generate U tilde and V tilde from Cox model, and we generated the dependence structure from a Clayton copula. The truncation time A tilde is generated from an exponential distribution, and we consider two covariates, including one Bernoulli distribution and the other one from uniform distribution. To mimic the biased sampling mechanism, we pose some constraint that any data with U tilde plus V tilde greater than A tilde will be included into our study. Otherwise, the observation data will be dropped. And under this setting, the truncation rate is around 65, which means 65% of patients will be truncated. And here are simulation study results for two scenarios under different censoring rate. And for the proposed method, we can notice that the empirical estimate is very close to the true value. And also, the model-based average standard errors are very close to the empirical standard deviation for the parameters of interest. And for comparison, we also conducted the analysis using the naive method, which is we just ignore the biased sampling and analyze the observed data. And for the results from the naive method, we can see that it yielded larger biases, and it got switched a little bit. But the right numbers here belongs to the estimated cumulative hazard function from the naive method. We can notice that the cumulative hazard function from the naive method is severely underestimated comparing to the proposed method. And the next figure here shows clearly about that. This is the cumulative baseline hazard function from the proposed method and also the naive method. For the proposed method, which is the blue solid line here, it is very close to the true curve, which is the red line here. But on the other hand, the black solid line here represents the estimate from the naive method. We could see that the estimations from the naive method will be severely underestimated. And then we applied the proposed method to our motivating study, which is a cohort include 386 patients aged 65 years old or older who had cancer recurrence. And our covariates of interest include race, age, HR status, and also disease stage, AJCC stages. Our goal is to study the effects of the risk factors on the residual time, to study the median residual survival time, and also estimate the dependence structure between the two bivariate survival time. And we use a Clayton copula to model the dependence structure, and we assume a Weibull distribution for the truncation time to allow more flexibility. This table shows the estimated hazard ratio and p-value of the proposed method. From the proposed method, we can see that the elder age, advanced AJCC stage, and HR positive status are significantly increase the hazards. But on the other hand, if we just use naive method that ignores the biased sampling, the AJCC stage is not significantly correlated with the events. And also from the proposed method, our alpha from the Clayton copula is 0.10, which suggests a mildly positive correlation between the bivariate survival time. And also we showed the survival plot along with the 95% confidence interval for the proposed method and also that from the naive method. Similarly, conclusions as the simulations, we can see that the red curve represents the proposed method, while the black solid line represents the estimate from the naive method. And we can see that using naive method, the estimates of the survival will be overly estimated. And also for patients with profile, for example, age greater than 75, HR positive status, the median survival from the proposed method was 25 months, while it was 45.5 months from the proposed, from the naive method. So the differences were huge and will affect our conclusion on the survival. So this is a summary of this presentation. In this work, we proposed a bivariate survival model to explore the association between the risk factors and a time to event outcome from a prevalent cohort. And our model's assumptions include a copula model to model the dependence structure between the bivariate survival time. For the marginal distributions, we use Cox models to allow some flexibility. And also, we assumed a parametric model for the truncation time distribution. After that, we proposed a modified EM algorithm to improve computational efficiency. In the E step, we will have a closed form. And in the M step, we can use existing softwares to get the estimates updated pretty quickly. That's it. Thank you.
Is this on? Questions from the audience? Thank you very much.
Thank you.
Michael.
Thank you. It's a very impressive work.
Thank you.
I was wondering if can it be extended to incorporate variables, time-dependent variables measured along the ways? For example, variables measured at the time of recurrence in terms of severity.
I think so. If we will incorporate a time-dependent covariance, we may need to update the complete likelihood by plugging in the time-dependent parts in it. So I think it's doable. Yeah. Mm-hmm.
About the biased sampling, if I understood this correctly, it is the truncation distribution that addresses that.
Yes.
Which makes me ask the question about sensitivity and assumption, because it seems kind of analogous to ignorable versus non-ignorable response or non-response or whatever it is. Mm-hmm. So is there a sort of assumption there that cannot be verified? And
yes.
And how sensitive are your conclusions to different specifications for that truncation distribution?
Yes, there indeed has some identifiability issues. So that's why we assumed a parametric model to model the truncation time. And we don't show it here, but we performed some sensitivity analysis to evaluate if we assumed a wrong distribution, what would happen. And from that simulation, our suggestion is that we can use very comprehensive parametric models, for example, generalized gamma distribution with 3 parameters for the truncation time. And we evaluate some performance, and our conclusion is that if we assume a really comprehensive parameters, the performance is okay, even though the data may generated from Weibull or exponential or many distributions. Yes, so the best we can do is use comprehensive parametric models, because we cannot assume a semiparametric models. For the bivariate survival time, we use the Cox proportional hazard function, which allow non-parametric part for the baseline hazard function. So if we have two non-parametric models, that will cause some identifiability issues.
Thank you. Other questions? Yep, there's one there, and there's a mic there.
Hi, thank you for your talk. I was just wondering, two questions. Is it completely obvious how to choose a copula function? And if not, do you have any practical advice about whether it matters a lot to choose a copula function?
This is a good question. For the copula function, usually there are many selections, and if you use different copula model, we may get different conclusions. So in practice, we would suggest we can draw a figure of the cross ratio. That is, usually people will use that cross ratio figure to help select a proper format of the copula model. For example, if it's constant, it may suggest a Clayton copula. And we also perform some simulation studies to show that under biased sampling mechanism, we can still use that method, although we can see the trend. From the trend, it could help us determine which copula to select, even though the estimated cross ratio was not accurate. But the trend will tell us some information for that selection. Thank you.
Thank you very much.
Thank you for the nice introduction. My name is Yayuan. I'm from the University of West Ontario in Canada. This is my first faculty appointment. Before I joined the Western University, I have spent 2 years roughly in MD Anderson Cancer Center, the same institution of our first speaker. This work is one part of my postdoc research from MD Anderson, and it is the joint work with my two supervisors, Dr. Liang Li and Dr. Xuelin Huang. Now, let me start my presentation. Firstly, I want to start with a motivating example. This is an African-American study of kidney disease and hypertension. We call it ASK data. In this data set, patients with advanced chronic kidney disease are often enrolled in the study, and they are monitored through frequent clinical visits. Here, the time origin is the randomization time. From there, patient will come to the clinic to see the physician to do some lab tests and some physician examination or evaluation. I use this graph to illustrate the scenario we have. TI1 to TI2 to TI6 are the 6 intermittent observation time of the longitudinal data before the event occurrence. And I think some other personal demographic information are collected at the baseline. What we are interested in is the risk of end-stage renal disease or death. This is the event occurrence. I use the cross to denote in the graph. So we try to predict, for example, we try to predict the risk of the renal disease event or death at the most recent clinical visit, TI6, using the longitudinal information collected up to TI6. This is what we call the dynamic prediction. We include 3 time-varying biomarkers, eGFR, the logarithm of urine protein, and blood pressure. And we also have one more time-fixed variable, age. Here I just plotted the personal longitudinal trajectories of the eGFR and the logarithm UPRO of 20 randomly selected patients from the ASK data. I use different color to represent the different patients. Here we can see some issues of the longitudinal data measurements. Firstly, we can see the measurement time subject specific. The timing and the frequency of the longitudinal data observation vary across subjects and also over time. And we also can see the longitudinal trajectory diverse substantially across subjects. For example, some patients has a linear trajectory and some patients have a nonlinear. And some patients' trajectory are relatively steady, and some others, they may have a very sharp and instantly shortened variability. So these issues make the prediction at an arbitrary selector prediction time difficult, because at a selection time, for example, here I think this label is wrong. For example, at the 10th year, we want to predict the risk of renal disease event or death. We hope all the patients have measurements of longitudinal data at the year 10. However, we don't have, we may not have the measurement from all subjects. One way to solve this issue is we can carry forward the most recent observation to the time we want to do prediction. However, if the gap between 2 visits is wide, it means the visit time or observation time is too sparse, or if the longitudinal trajectory vary too substantially in shortened period, then the last value carry forward method does not work well, because the estimation and the prediction has some notable bias. Another way to solve this issue is we can specify a parametric model to model the longitudinal data. However, because of the larger diversity at an individual level, it is hard to specify a reasonably good model to model the longitudinal data, especially if we have multiple longitudinal variables, just like the example from our ASK data. It is basically impossible to correctly specify all the models for longitudinal data. So later I will introduce a landmark model method, which could be used to flexibly model the longitudinal data and estimate the model parameters non-parametrically. About the prediction of event risk in practice, one way is called static prediction. This is still the most widely used method in practice. Static prediction means the information collected before or at the baseline is used for predicting the subsequent clinical event. This kind of prediction method does not work well in longitudinal context, because we didn't incorporate the history of longitudinal data into prediction, and we didn't even adapt to the progressively changing risk set. Dynamic prediction, on the other hand, is more powerful in longitudinal context. It uses the prognostic information collected up to the time of prediction into the event risk prediction. And this kind of prediction can be made in a real-time fashion at the patient's clinical visit to the physician's office. And here I use the same plot to illustrate what dynamic prediction do. For example, at the most recent visit, TI6, we could be interested in the renal disease risk in the future 3 years. This is a 3-year survival past the time I6. So we want to use all the information collected up to TI6 to predict the 3-year event risk in the future. Now, about the dynamic prediction, there are 2 main strengths of methodology in literature. The first method is called the joint modeling method. Joint modeling method typically specifies submodel for the longitudinal process and the survival data process, and use some shared random effects to account for the inter-process correlation. Another method is called landmark modeling. Landmark modeling specify a model for the residual event time given a selected landmark time, and use the observed longitudinal biomarker history collect up to the landmark time as predictors in the model. I want to say joint modeling method has a typical problem because we need to specify, usually people specify parametric models for longitudinal data, and they have a full likelihood which includes many random effects, and those random effects are unobservable, so we don't have a closed form for the, usually we don't have closed form for the full likelihood, and we need to use some numerical method to do the integration over random effects, making this method very computationally intensive. Landmark model, on the other hand, is very computationally easy, and also could do flexible modeling of longitudinal data. A comprehensive discussion about the landmark models and the joint model can be found in this paper recently published, I think published in Biometrics, I think. They did some simulation study about both of these 2 methods and highlighted the strengths and the drawback of each method. Now, let me quickly introduce some notation that I will use in the following slides. TI is the time to the clinical event for subject I. CI is the censoring time. And in survival analysis, typically we can only observe the minimum between them, which is called observed time. And the delta I indicates the fading or censoring status. S is the selected landmark time, and Ti minus S is the residual event time. We define the landmark model based on the residual event time. We could have multivariate longitudinal data. I use the set IT denote the vector of longitudinal variable measured at time t. And the TI1 to TIMI are the MI intermittent observation times of the longitudinal data prior to the event occurrence or loss to follow up. The event risk is defined by the probability of an event occurring between the selected landmark time and the landmark time plus prediction horizon, given the survival up to the landmark time S. So what we want to estimate or we want to predict is this conditional probability, which is the event risk. Okay, because our model based on the linear transformation model. The traditional linear transformation model, which has this form, the G function G is a link function, is a specified link function. The S of t is the survival probability, survival function of the event time. HT is the non-parametric component. Theta is the vector of covariance, and beta is the corresponding coefficients. This model can be written in a linear additive form presented by equation 2. Sorry, I think I don't know why here the screen shows differently compared to my computer. Here should be the Ayrton axiom. The axiom Ayrton in the equation 2, the distribution of the Ayrton corresponds to the link function. So if we specify the distribution for the Ayrton, we actually specify the link function for equation 1. Now, we consider a parametric specification or the parametric transformation in the linear transformation model. The Hesse function of the Ayrton is specified by equation 3. The parameter R, we consider it as some real number like 0, 1, and 0.5, 2. Particularly, if R equals 1, it corresponds to a proportional Hesse model. If R equals 0, it is a proportional Cox model. If R equals 1, it's a proportional Oz model. Then the paper we published in JRSSC recently proposed a landmark linear transformation model presented by equations 4 here. This model specified the linear transformation model for the residual event time and allow all the model parameter to vary along the selected landmark time S. Actually, we also allow the link function G to vary along S. However, we didn't find a real data example can illustrate different link functions at a different landmark time. Even in our real data example, ask data, as a result, when we select the link function at different landmark times, it turns out that the proportional Oz model is globally the best prediction model. But if someone has the example, if someone has the real data example which leads to the different link function at different times, I think there would be a very good opportunity to show our method is particularly appealing, more appealing than other methods. Then let's talk about the estimation of model parameters and how to conduct prediction. If we have measurements of the longitudinal data at the selected landmark time S, then event risk can be predicted based on this equation with the model components H, HS, and beta S plug in. However, because we allow the longitudinal data to be measured at intermittent, actually sparse measurement time, so we cannot have the prediction, the predictor variable set I as to be observed for each subject. However, but we hope we can have the prediction feasible at any arbitrary time, because considering a new patient could come to the physician office consulting about the prediction of future event risk at a new visit time, TIJ. This visit time could be different from anyone of our original sample. Now, one way, as I mentioned earlier, to do that is use the last value carry forward method. That is, we carry forward the most recent observation of longitudinal data into the prediction time. This is an approximate way to do the prediction, and of course, it leads to some bias. And especially when the measurement times are sparse and biomarker have a large short-term variability, this method, later I will show from our simulation, leads to obvious bias and inaccurate prediction. We now discuss how to do the estimation of the non-parametric component and coefficient at an arbitrary select landmark time in the situation of subject-specific measurement times of longitudinal data. We propose to use local polynomial smoothing to group the measurement time of longitudinal data in the neighborhood of the landmark time and smoothly estimate beta and h. I don't know why here it shows very differently from my computer. So I don't want to explain much about the equations. This is a standard way to include a local polynomial smoother in the estimating equations. Here the kernel, yeah, the clicker, even the clicker doesn't work. The kernel weight kh, it includes a kernel density function and a bandwidth sequence. The yi and I are the typical counting process notation defined for the residual event time. And theta I and theta I, those two notations are defined following Taylor expansion. Now, the estimation of the theta s and h us can be obtained by solving these two estimated equations using an iterative algorithm similar to that introduced in Chen et al. In 2002. Now, let me quickly introduce some simulation results. Our data were simulated directly from the landmark linear transformation model. And this is not a trivial way to simulate data, because usually people generate data from joint model. Then they marginalize over random effect to obtain a marginal model. If the problem with that method is usually they cannot obtain an exactly they cannot obtain exactly correct landmark model. So the performance and the estimation could be performed based on misspecified model. Here we have a paper particularly discuss about the relationship between joint model and landmark model, and propose a way to generate data directly from landmark model. I need to quickly go through it. Here I just want to present the result from proportional odds model because it's close to the real data example. And we generated data basically similar to our real data example, like the sample size, the frequency of longitudinal data, and the number of time variant predictors, everything very similar to our real data example. And we use the mean square error to evaluate the calibration of prediction, and we use AUC to evaluate the discrimination ability of prediction. Now, again, here, sorry about the messy. Now here, we can look at, I highlighted the result from our proposed method in blue color, and highlight the result from last variable carry forward, which I consider as a naive method in red color. Here we can see the MSE. Our method produced a much smaller MSE than the last variable carry forward. So the calibration ability of our proposed method is much improved. Although the discrimination AUC, actually, they are pretty much close to each other. We also consider the parameter estimation of the regression coefficients. And we see here the last variable carry forward method, they leads to very large bias, and our method reduced that substantially. Now, quickly, let's talk about the application to the ASK data. We have roughly 1,000 patients from the ASK data. Among them, almost a half reached the terminal event before lost to follow-up. And follow-up is up to 12 years, and each patient has roughly 9 follow-up visits. And we include gender, gender is the fixed variable, and we also include EGFR, logarithm of urine protein and blood pressure, and the slope of EGFR and the volatility of blood pressure in the past 3 years as predictors. What we are interested in is the 3-year survival at a selected landmark time for prediction. There are a couple of things I considered as a tuning parameter, and we selected the tuning parameter data adaptively using Monte Carlo cross-validation. One is the bandwidth in the kernel smoothing. Another one is the parameter R in the link function. Especially just, let's look at this plot. This plot shows the AUC versus different value specified in the link function. And we see at S48 months and 72 months, the R equals 1, which corresponds to the proportional odds model, is the best model, which gives the highest AUC. And this project just simply shows what dynamic prediction do to predict a personalized risk of clinical event. Here I use 48 as a landmark time, which is denoted by a vertical dashed line. What we try to use, on the left of the vertical dashed line is the longitudinal trajectory of 3 longitudinal biomarkers, and on the right-hand side is the prediction of future survival. We try to use the data, the information collected up to the 48 months to predict the future probability of survival from the renal disease event and all deaths. This is the ROC plots of our proposed method and the last very carry forward method. Our method produce AUC basically all higher than 0.75, so this is a very good prediction. To conclude, here I talk about the dynamic prediction of a clinical event with longitudinally measured biomarkers. We assume the biomarkers are specified, sparsely and intermittently measured. A landmark linear transformation is proposed to model the residual event time. We use the local polynomial smoothing to estimate the model parameter smoothly and non-parametrically. So we impose the minimum of model specification for the longitudinal data. The proposed approach can be easily used to analyze multiple longitudinal variables. Here, especially, I consider 3 to 4 longitudinal biomarkers in my simulation and real data example. And we see the computational burden does not increase with the number of longitudinal variables. And this is, I think, the biggest advantage of our method compared to joint modeling method. Okay, I think this is the end of my presentation, and thank you so much for the attention.
Thank you very much. And now we have time for a few questions from the audience.
Thank you for the excellent talk. I was wondering how the method handles informative observation times. I think you gave an example with non-informative observation times. Just curious about the other case. Thanks.
Yeah, this is a good question, and actually related to my PhD work conducted in Waterloo University in Canada. At this stage, we assume the observation times of longitudinal data are non-informative. But it is possible to extend it to informative observation times. Actually, in my PhD research, I look at the informative observation times for interval censored survival data. And similar, I believe there's a way to extend the dynamic prediction to incorporate the informative observation times. Yeah, thank you.
So I'll have one. Just to wrap up, just to step a bit back from the specifics of the method. So apologies if it is a slightly unfair question. Thinking about the uses of this, because in terms of medical statistics, this represents a slightly different point of using statistics for medical research. So this is not about discovering causes of the disease. This is predicting with known causes. So from what you know, from your experience, what would be the use of these models in clinical practice? What do the clinicians, the doctors, do with the prediction for an individual patient?
Yeah, I want to say this is a question I answer a lot of time to my collaborators from our medical school. Yeah, I believe this method is a very good way to do the personal, we call it individual risk prediction, right? At any post-baseline time point using the longitudinal prognostic information. And this can be used for early detection of disease progression, and also can be used to select the best treatment, what we now call it personalized medicine, right? Precision medicine. We try to do some work to reach that goal, to select the most appropriate treatment for patient using their prediction of the future risk. For example, if we have 2 patients come to the clinic and they have a different history record of the disease progression, and we want to use this information in the prediction, and we can identify the patient who have highest and who have the most severe disease progression status. And this kind of patient could be have more priority for the treatment. Yeah.
Hi, thank you very much for a very interesting talk. I was just wondering if the number of observations for per patient is important in the fitness of your model. So your ROC value change depending on the at least number of the observed time per patient or not. So for example, if you have many patients with 2-3 observation time only, and then few like with 9, 10, even more. So have you looked at it?
Yes, we need to assume each patient in the sample has one measurement at least. Usually in medical practice, the baseline measurement is available. So this is the minimum information we can use. Actually, this is the same for last very carry forward. If we don't even have the baseline information or we don't have any measurement, we cannot do the prediction for this patient. So we assume we have at least one measurement of longitudinal biomarker for each patient from the sample. And the frequency, and for example, the number of measurements of the longitudinal data, yes, it has some impact in the estimation of a parameter and or the regression coefficient, because we use the kernel smoothing method. So the bandwidth selection actually is affected by the frequency of measurements. Generally speaking, the bandwidth will be smaller if the measurement number is larger, right? There's a trade-off for, however, the bandwidth selection. As I remember, if we have a smaller bandwidth, we have a small bias but a larger variance, like that. So, but it still depends the quality of data collection, and bandwidth could be selected data adaptively. Yeah.
Thank you.
Okay, thank you very much.
Yeah. Thank you so much.
Thank you for the opportunity to speak. I'll be presenting on, it's a paper called Adaptive Design in Surveys and Clinical Trials, Similarities, Differences, and Opportunities for Cross-Fertilization. And this is joint work. I work on adaptive trial designs, and I'll tell a bit about my research in a moment. It's joint work with Peter Miller, Michael Thiem, and Ben Reist, and they're all with experts in survey sampling. Elizabeth Stewart, she works on clinical trials with me, as well as Tom Lewis. He has a foot in both camps. So the idea was we joined forces, clinical trial statisticians and survey sampling statisticians, all working on adaptive designs in our respective fields, and we talked about how might we learn from each other. So that's what produced this paper. A disclaimer that views presented are all my own, not necessarily anyone else, including the FDA and the European Medicines Agency and any other organization. And this is the link to the paper. Before I present on the paper topic, I wanted to give a one-slide overview of my research in case anyone might be interested in collaborations. I love to talk about any kinds of collaborations. So my work is on developing improved statistical methods and open source software for the design and analysis of clinical trials. One area I work on is statistical methods for reducing the sample size required, or increasing precision and reducing the sample size needed by optimal adjustment for prognostic baseline variables. I think this is one of the most underutilized techniques in clinical trial analysis and can lead to big improvements without making any model assumptions. Another area I work on is new adaptive enrichment designs. These are designs where there's a pre-planned rule for modifying patient enrollment based on accruing data in an ongoing trial. And the goal is to maximize power under cost and sample size constraints. And I have my research group, we have an open source software tool for trial planning and optimization with clinical applications in stroke, cardiac resynchronization devices, Alzheimer's disease, and HIV treatment. If you're interested in any of those, you can go to my website. You can Google Michael Rosenblum. You have to skip over the first Michael Rosenblum, who's not a statistician, but it's someone, he's a journalist. And actually, we're friends on Facebook, this other Michael Rosenblum. And a quick story, an aside, is that once my wife noticed that my mother was liking everything that this other Michael Rosenblum was posting up there, and then realized, oh, my mom thinks that's me, this more famous Michael Rosenblum, who's a journalist. Though somehow our political views are almost identical. But that's an aside. And the third area I work on is stress testing clinical trial designs. So the idea, and this is funded by a grant from the Burroughs Wellcome Fund, and it's developing software tools that systematically probe a trial design. This is before the design is implemented. Probe for weaknesses, specifically weaknesses to small violations of assumptions that go into a trial design, and to try to fix the weaknesses before the design is conducted. And so the idea is we stress test people by having them run on a treadmill and seeing what does that trigger in terms of cardiovascular output. We stress test cars by smashing them up at different speeds to see how well they protect a potential person. Stress test banks by going in and seeing what would happen under a hypothetical downturn in the economy. And I'm just trying to apply that technique, stress testing trials before they run to try to avoid potential preventable problems. So going back to the main topic, the paper that I'm presenting, first I'll talk a bit about trial designs and then survey sampling, and then talk about 5 different potential ideas where techniques from one area could be cross-fertilized into the other. So adaptive trial designs, why should one be interested at all? So as an example, pharmaceutical companies are interested. So the Clinical Trials Advisor, this is a newsletter. This is back about 10 years ago. 10 years ago today, it looks like. There was an article that said adaptive trial designs save Merck, the pharma company Merck, millions. And I blew up the first line. The first line says that an adaptive clinical trial conducted by Merck saved the company $70.8 million compared with what a hypothetical traditionally designed study would have cost. Now that, I was very surprised, because I work on adaptive trials. I was surprised how you could save that much money. And it turns out, well, does anyone have any ideas on how you could save that much money? Just take a guess. You can just shout out an answer. Earlier stopping, that's right. And then to take it further, how could you save 70.8 million? It's a very precise number. 70.8 million from early stopping. I mean, a typical trial, the costs for a phase 3 trial could be maybe 10 million up to, some trials do cost more than 70 million, but that's pretty rare. Sorry? Exactly. So this 70, it's very good. It's very rare that someone in the audience guessed this, but that's exactly right, that the 70.8 million comes from, they estimated that if they could have stopped the trial 2 months earlier, that would have led to 2 more months on patent for their drug, and a blockbuster drug they assumed would bring in about 35 million per month. So that's their calculation. I don't have any connection to Merck or any pharma companies. So it turns out, actually, I should have stated that it turns out that this line here is not true. When I looked into it, it really should have said an adaptive clinical trial conducted by Merck, a hypothetical adaptive clinical trial, looking back in retrospect to see what would have happened had they used an adaptive design. It would have saved, they project it would have saved 70.8 million. And I apologized kind of at the end of I was presenting this at a talk for new trial designs for HIV treatment, new antiretroviral drugs for treating HIV. And I said, I hope there's no one here from Merck who I'm insulting. And it turned out the next speaker was from Merck. And I would have known that if I just looked on the list of speakers. It wasn't a surprise. And I apologized to that speaker afterward, and he claimed, this is anecdotal, but he claimed that at Merck at the time, this is about 10 years ago, if you don't propose an adaptive feature to your trial design, then you have to explain why not. But that's really the opposite of how it should be. In terms of my work on adaptive trial designs, I think you should only use an adaptive design if there's a clear benefit to doing an adaptation. Otherwise, you should stick with a simpler design. And so a quick overview of adaptive clinical trial designs. So the key thing, and this is that in bold here, that adaptations in clinical trials need to be protocol-based. It has to be based on a pre-planned rule. It's not just do whatever you feel like when the data came in. So there's a pre-planned rule for modifying features of the trial design as data is accruing. And there are guidelines on how to do this put out by the European Medicines Agency and the US FDA. Some of the most common examples of adaptations in trials are early stopping for efficacy, futility, or harm. The focus here is phase 2 and 3 clinical trials, so the more confirmatory type clinical trials. And so early stopping is sometimes called group sequential designs. Three other examples of, by far the most common is that first one, group sequential designs. But also somewhat common are sample size re-estimation, so changing what the total sample size will be as some data has accrued. Maybe the variance was under or overestimated to begin with. Dose selection, you have multiple doses, and you might start dropping arms with different doses. And also biomarker adaptive designs, and that means that a change is made in terms of, based on estimates of the treatment effect in different subpopulations. It could be based on a biomarker, such as a genetic marker or disease severity at baseline. And so on the top of this slide, so I'm going to transition from trials to surveys, sample surveys. So on the top, there are three bullet points, or ABC, describing different trial adaptations, some of which I already mentioned. I'll just mention the new ones. So there's modifying enrollment criteria. That's called adaptive enrichment. There's changing the follow-up time and changing the randomization probability or endpoints in a trial. And then when we go down to surveys, so in survey sampling, the bottom half of this slide, the types of adaptations are quite different. So here are some of the examples of adaptations in a survey would be changing modes of contact or data collection. So you might start off with some of the cheaper forms, either phone calls or mail or online, and then follow up if people don't respond with an in-person visit, which is more expensive. You might be is modifying timing or frequency of contact attempts. Another adaptation is changing incentives, giving money, more money, or other incentives. And also D is deciding when to stop data collection. So really the one common feature is the when to stop rules. But they're quite different. And I want to mention, so with a focus now on survey sampling, something that will come up later in the talk is the idea of balance or representativeness. So I use those interchangeably, and they're measured. There are different ways to measure balance within a trial, and really, or representativeness. What it's referring to is similarity between the distribution of auxiliary variables, that's variables that are known to the survey staff before any contacts have been made, so base information. Similarities between what that and those auxiliary variables over the target population, in the sample, and the subset of the sample who respond. So it's basically looking at, is there some imbalance related to non-response? And these are measured by there are different ways of measuring it. We focus here on R indicators. And the R indicators, they potentially identify which variables contribute most to the sample imbalance, and also the direction of misrepresentation. So you might have an overrepresentation of men or different age groups in the people who respond, which would potentially cause bias. The goal is to try to adjust the sampling, so modify the trial to try to improve those measures. So here are the 5 ideas. I'll go through relatively quickly, just an overview of the 5 ideas that we proposed for cross-fertilization. The possibility of cross-fertilization is there because of shared goals among trialists and survey samplers of minimizing costs while making valid statistical inferences in the presence of non-response or missing data. And some common themes throughout these are that adaptive methods developed for surveys, they could be applied to improve external validity and sometimes also internal validity in trials. And in the other direction, adaptive methods from trials, they might be applied in survey sampling to improve internal validity and efficiency. So each of these 5 examples, there'll be an arrow at the top. So this is an example from surveys to clinical trials, taking ideas from surveys, applying it in clinical trials. And the idea is to systematically monitor representativeness and improve it by targeted enrollment and follow-up in clinical trials. So the idea is you might be able to use this for 2 purposes. The first is improving internal validity in the trial. The idea is compare baseline variables, pre-randomization variables, of the respondents to those of the overall sample. So this is looking at, look at everyone for whom you have the outcome measured versus people who are missing their outcome, people who dropped out of the trial, and compare baseline variables to see if there are large discrepancies between them. And then try targeting intensive follow-up. So there's something called double sampling. You can make extra efforts to go after people who dropped out and try to get their data, but you can be selective in who you go after to try to get that data from, and try to target the people who will improve balance or representativeness the most. And you can use R indicators to do that. And you can use, you can do the same kind of thing for improving external validity in the trial. If you have a target population and you know from a registry, for example, the distribution of baseline variables that you'd like to have, and then you see in your trial, here's who I enrolled in my trial. I have too few people with high severity of disease or have too few people with a certain biomarker or certain demographic characteristics, you can try to change your enrollment intensity and try to get more people in the trial who have the, to try to get balance that's closer to your target population. It will help you with generalizability. A second example in the same direction of transferring techniques from survey sampling over to clinical trials is collection and use of paradata to improve retention and protocol compliance. So paradata, this was new to me when I was learning about survey sampling. Paradata is contextual information collected alongside or before the outcome data. And so here's some paradata examples in what it is in clinical trials, what it could be. So these are things collected at or before clinic visits. So things like the number of attempts needed to schedule a visit with a participant. Does the participant tend to arrive early or late? How many questions they answer in surveys. So when they come into the visit, either having an in-person or a computer-based survey, asking information to how many questions are answered, what's the delay time in answering the questions. Especially with a computer-assisted survey, you can collect lots of information. And also clinician observations during visits of participant satisfaction or dissatisfaction with the study. And the idea is to use this sort of side information, stuff that is just collected, it's not the primary goal, but you collect it on the way, this paradata to predict who's likely to drop out of the trial, and also people who might fail to follow the trial protocol. And then use that, and you could also use clinical information in addition to help you do this prediction. And use that to help you target with interventions to target who is likely to drop out or likely to not comply with the trial protocol, and try to do interventions to improve that, to keep them in the trial. You might change incentives, you might change, or maybe just have meetings with those individuals to talk about what might help keep them in the trial. So going in the other direction, cross-fertilization, so taking ideas from clinical trials and applying to surveys is looking at stopping rules, early stopping rules. So surveys are often conducted in waves of data collection, and there are sometimes are pre-planned rules for when to stop data collection, but based on accrued data. Sometimes can be more ad hoc. But there have been proposed stopping rules by Rao et al. And Wagner and Raghunathan. These focus on reducing non-response bias, and how they work is they do some kind of imputation, and in order to predict the probability that the next wave of data collection will lead to a change by a certain amount, a minimum threshold, in the estimates for key variables. And if it's a high predicted probability that there will be no change based on the next wave of data collection, then the trial is stopped. And in contrast, the clinical trials also have stopping rules, but the goal is typically different. The focus is on achieving a desired precision and power. It's much more focused on variance than bias. And there's general techniques called information monitoring that are used, especially in large trials, phase 3 trials. So the proposed idea is to compare both types of rules in simulated adaptive surveys and look at bias, variance, number of contact attempts, which look at the cost, and see whether some of the techniques for early stopping in trials could be useful, or could even be hybridized and combined with some of the techniques already in use in survey sampling to try to improve, to get the optimal trade-off in terms of cost versus bias and variance. And another idea that could be taken from clinical trials and transported over to survey sampling is called sequential multiple assignment randomized trial designs, or SMART designs is the acronym. And the idea is that in each wave of the survey, non-respondents get randomized to different intensities, different contact modes, different incentives, and to respond. And the goal is to learn basically what's the optimal set of modes of data collection and the optimal sequence. So if what's the best first line mode of data collection, for example, phone calls or mailing. And for people who don't respond, what's the best second line technique to use? And this has been done. There are examples of this already in the survey sampling literature. Dvorak and Chang did this. But the proposed idea is to there's a lot of literature and methodological advances in the clinical trials community and causal inference community on estimating optimal sequential treatment rules within strata of baseline variables. And that could be ported over to the survey sampling world, where you use estimation techniques from Susan Murphy and Jamie Robbins and Mark Vanderland and Alex Ludka are just some examples, techniques to estimate the optimal rules, which can be tailored to participants, their baseline characteristics, to try to find what's the best sequence of modes of survey sampling to apply to minimize cost. Okay, perfect. To minimize cost and maximize and minimize bias. And there's one more but I'll skip over, and I'll leave that for if you're interested to read the paper. That'll be I have to leave some mystery. And just a quick caution or warning. A caution and warning is that it's important to do simulations and careful assessment of any kind of adaptive design before you run it. You have to do simulations and look at what happens under violations of your assumptions. So that applies to all the techniques here in both survey sampling and in clinical trials. And I'll just end by saying that the analysis at the end of any adaptive design, whether in trials or surveys, you have to take into account that there was an adaptation. You can't just assume that whatever trajectory was taken, that always gets taken. It needs to be factored in at the end into the analysis. Thank you very much.
Thank you. Slightly behind time, but we do have time for questions. Does anyone have questions from the audience? I will do the honors for the mic.
In the transition from surveys to trials, some of your suggestions were essentially pre-randomization. Others were post-randomization and added targeting or added response effort to get a patient to stay in the trial is an intervention in addition to the treatment which was randomized. And so I think one has to have particular caution about interventions after the point of randomization.
Yeah, that's a great point. I mean, adaptations, especially any adaptation that would impact a participant after they've been randomized, I would say you have to be very, very careful. I 100% agree. And the types of adaptations I'm talking about are essentially trying to get increased intensity of attempts at follow-up. And that, it's true, you have to be very careful with that. I mean, if that could influence, it could change. The goal is actually to change the distribution of your estimator because you're trying to get data that you otherwise would have had to impute or try to deal with in another way. But I 100% agree. Any adaptation that involves something post-randomization, you have to be extremely careful in how you do it.
Although we're over time, I can't resist asking one more.
Yeah, please.
Slightly more broadly again. So this was about one specific contact point between survey sampling and randomized experiments. There could be others if we see this bigger picture that randomization is in effect probability sampling of potential outcomes, to put it that way.
Yeah, exactly.
So in your discussions in the nicely cross-disciplinary team, so to speak, cross-methodological team, how much do people appreciate these connections? And in other contexts, other than adaptation, how much of this kind of work goes on? Do you know?
That's a great question. I can only give kind of essentially an anecdotal answer based on my discussions with within this group. And I have to say, this paper really was a joint effort by everyone, all of the authors. My impression is that there is potentially a lot to learn from both sides, and that there's, at least I can say on my own side of being on the clinical trials side, there are many ideas from survey sampling that have been stolen already. Stolen is maybe not the most fair word, but the things like double robust estimation, I mean, that was actually came from the survey sampling world, was done earlier in the survey sampling world. But my impression is that we, at least in clinical trials, there's a lot we can learn from survey sampling, especially about how to targeted interventions to deal with missing data, to deal with people who drop out. So I guess that to answer your questions about going beyond adaptive designs and just saying what, in general, survey samplers versus clinical trials, do the communities have a lot to learn from each other? And I'd say yes. And I think my impression is there is not a ton of communication between those worlds. A little bit, but I think we would both, at least clinical trial side, we would benefit a lot from learning more from survey statisticians.
And vice versa, I'm sure. Thank you very much, Michael. Thank you to all of our three speakers and the audience. If you want to go to one of those rapid fire talks, then you'll have to move fairly rapidly now, I'm afraid. So, but thank you again.